So Erin, why don't we get started and people will continue to join.
Okay.
So it's a pleasure to introduce Erin Hazlett to our virtual psychiatry lecture series. Erin, as she'll tell you probably, came to Mount Sinai to work in the Bucksbaum lab and in the Seaver lab, but it's a different Bucksbaum and a different Seaver. And she's been here since 1993, and she's really been a leader in understanding kind of imaging and neurophysiology and neuropsychology of schizophrenia, schizophrenia-related disorders, affective disorders, personality disorders generally, and then most recently also being very active in suicide and suicide prevention. Erin I've known her for years, and she's very productive, very well funded, and really does, I think, a lot of critical work in these domains of trying to understand what's going on in the CNS and major mental illness, and really kind of pioneering multi-measure approaches and using them as biomarkers or predictors or stratifiers, or to understand the underlying biology for some of these very significant disorders. And today she'll talk about neurobiology of personality disorders and talk about neuroimaging and psychophysiological approaches. As I said, two of the methodologies that she's really pushed forward in the field. So Erin, thank you for doing this.
Thank you.
And Cheryl, thank you for joining. I know you were here yesterday too. I see that Cheryl's on.
Sorry, Cheryl. I had to tell Joe that story. I can relate to it very well. All right. Well, thank you for that kind introduction. I'm really happy to be talking to my colleagues for the most part. I want to start by showing that I have a long list of collaborators over the past 2+ decades, and I want to give a big shout out to Larry Seaver, who was one of my mentors when I came in 1993 as a postdoctoral fellow. Larry just passed away recently, and we're all very sad about that. We've been missing him for a few years because he had a serious thalamic stroke, but he recently passed, and this is going to be a little bit of a tribute to him. I have other collaborators at the VA, at the MIREC. You can see here many of them, and the mood and personality disorder group as well. It takes a real village to do neuroimaging research. So I'm going to tell you today about 2 lines of our ongoing research that Joe mentioned, actually. So one has to do with the schizophrenia spectrum. I'm going to start with that. And sort of our overarching question in this area of our research is, do people with schizotypal personality disorder have a protective brain factor that mitigates full-blown psychosis and the severe cognitive and social deficits that we typically observe in schizophrenia? So first, I want to also mention that there are many advantages to studying people with schizotypal personality disorder. This disorder is categorized as a schizophrenia spectrum disorder in the DSM-5. So it gives us key insights into schizophrenia. By studying these individuals, you can avoid confounds which plague schizophrenia research, like medication, long-term hospitalization, and severe psychosis. It's challenging sometimes to get some of the schizophrenia patients to do tasks in the magnet, much easier with schizotypals. We can have discovery of risk and protective factors, which I'm going to talk about today, for more serious mental illnesses like schizophrenia. And MRI studies are able to examine the pathophysiology and help develop treatment targets for schizophrenia spectrum disorders. So actually, this is a slide I borrowed from Larry Seaver from the New Yorker. This cartoon shows what looks to me like a person with schizotypal personality disorder on their birthday. And here I have listed the 9 diagnostic criteria for this disorder. You have to have 5 of the 9 to meet the criteria according to the DSM. And as you can see, this individual looks very lonely, lack of close friends, might be a little anxious. He's a little oddly dressed. And so these are some of the diagnostic criteria that characterize this disorder. It's important to point out that we've been talking about this disorder way back when, before we were all here. more than a century ago, Kreplin talked about people with eccentric personalities in the families of schizophrenia patients. He talked about, probably for the most part, this was to be regarded as latent schizophrenia. He concluded that such relatives probably suffered from a disorder that was, quote, essentially the same as schizophrenia itself. So he had a lot of insights back in 1909 about this disorder. So in the late 90s, early 2000s, we did a lot of very exciting imaging work at Mount Sinai. Monty Buxbaum, who was one of my mentors early on, we were using PET at that time, and we showed that and we were using cerebral blood flow as well. So higher cerebral blood flow in the middle frontal regions during the Wisconsin card sort in people with schizotypal personality disorder, and better task performance was associated with greater activity in the frontal lobe. I published a paper in the American Journal that was very exciting because it got on the cover of the American Journal in 1999, showing that glucose in the thalamus, this was using FDG PET, was actually normal in people with schizotypal personality disorder, but lower in the medial dorsal nucleus region, which is kind of in this area, in people with schizophrenia. And we also showed that there was higher glucose in the dorsolateral PFC in Brodmann area 10 during some verbal memory task performance, the version of the California Verbal Learning Test. So this work really led to this seminal paper that Larry Seaver and Ken Davis published in the American Journal in 2004 that talked about social deficits and cognitive impairments that are seen in the entire spectrum of schizophrenia, spectrum disorders, are likely the result of underlying genetic diathesis that, in conjunction with modifying environmental factors, adversely affects cortical structures, primarily the frontal and the temporal lobe. And this was sort of the figure that he had in the paper. But importantly, this paper discussed the idea that in schizotypal personality disorder, the frontal lobe acts as a buffer against deficits and dysfunctions. And this was really important work that sort of drove the focus of my research. So the idea that increased prefrontal cortical compensatory, I think my slides are on a timer, buffers could diminish the abnormalities seen in schizotypals relative to schizophrenia. So we were also doing structural MRI, and then after we realized that PET scanning wasn't as easy, or PET scanning was challenging, we started doing functional MRI. So our structural MRI studies showed that schizotypals actually had larger volume of parts of the frontal lobe compared to people with schizophrenia. So you can see here, larger volume in Brodmann area 10. This is relative to healthy controls at the 0 line. Pretty good size sample. And schizophrenia patients had a huge decrease in this region. So the idea of greater frontal reserves extended also to structural MRI, not just functional imaging. And then more recently, using a modality called diffusion tensor imaging, which allows you to look at the integrity of white matter tracks. Mark Lenner and I published a paper. He was a psychiatry resident, so this was exciting. And we showed that in schizotypals, they had less marked abnormalities in the corpus callosum, in the genu of the corpus callosum, relative to people with schizophrenia. You see a much more marked abnormality. So this is an important white matter tract because it is in the frontal lobe and actually connects the frontal lobe hemispheres. Also more recently, Chi Chan, who's an assistant professor at Sinai, showed some abnormalities in white matter in people with schizotypal personality compared to healthy controls. We didn't have schizophrenia patients in this particular sample, but you can see here that the schizotypals did have lower fractional anisotropy which is the measure that we use for white matter integrity. In white matter tracts, the sagittal stratum and the longitudinal fasciculus, the superior longitudinal fasciculus, which is really important in connecting frontal and temporal lobes. So they had lower FA compared to healthy controls. And it was primarily in the left hemisphere, which replicates a lot of the work in schizophrenia. Abnormalities are typically more pronounced in the left hemisphere. Just a little bit about some of our task-based fMRI work. Actually, a shout out to Harold Koningsberg, one of my colleagues at Mood and PD. So in 2005, Harold showed that people with schizotypal personality disorder had greater fMRI activation in the middle frontal gyrus during a visual spatial memory task. So greater than normal activation and no behavioral performance differences. So the schizotypals performed at the same level as healthy controls. So again, the idea that perhaps they're using their frontal lobe more than healthy people, and that allows them to compensate behaviorally. We published a paper in 2008 showing also enhanced bold activation in frontal striatal thalamic regions during a pre-pulse inhibition task, which is basically a psychophysiological paradigm that measures sensory motor gating, but it was an attention version where they had an attended tone and an ignored tone. And the schizotypals did have trouble ignoring the stimulus that they were supposed to ignore. And then the group at Harvard, Sean Lee Dickey, this was Bob McCarley's group, showed greater response in schizotypals, greater activation in the superior temporal gyrus during a tone discrimination task as well. So very consistent across labs that people with schizotypal personality disorder show this potential sparing compared to schizophrenia. A little bit about functional connectivity, which is something that is done more recently now, where you use a seed, and in this case, it's showing that maybe the seed is in the amygdala, and you look at basically its correlations between activation in different regions, and it allows you to look at the functional connectivity. So if your amygdala is activated, how is that correlated with, say, the frontal lobe? So recently, Phil Zesko and I have been working together on a large data set, and this is now under review. It's a lot of little slices here, but basically this is looking at functional connectivity, getting back to the thalamus, one of my favorite areas. We actually had seeds in different regions of the thalamus, different nuclei. So here, this is the analysis from the pulvinar seed. So the pulvinar is a nucleus in the thalamus that's connected very much to the temporal lobe, so of interest in the schizophrenia spectrum. And what we found was less than normal pulvinar to temporal connectivity in schizophrenia, which you see down here at the bottom. They're more yellow. So this is the connectivity between the pulvinar and the temporal lobe. So it's much lower in schizophrenia compared to the orange and red that you see at the top here in healthy controls. And interestingly, the schizotypals, which are shown in the middle here, are really in the middle. So their activation didn't really differ significantly from the healthy controls or the individuals with schizophrenia. So again, showing sparing relative to people with schizophrenia in this functional connectivity circuitry. Similarly, we looked at medial dorsal nucleus connectivity and found pretty much the same pattern. So relative to healthy controls, down at the bottom here, you see schizophrenia patients have much lower medial dorsal to middle frontal cortex connectivity. So the medial dorsal nucleus is very much connected, has reciprocal connections to the parts of the medial and middle frontal cortex. And again, schizotypals were intermediate, so not significantly differing from healthy controls or people with schizophrenia. And there were some clinical correlations there as well, but I'm not going to get into that. So kind of summarizing this and building on that seminal Seaver and Davis paper, we think that frontal lobe reserves and functional connectivity protects people with schizotypal personality disorder from decompensating into florid psychosis. And temporal lobe abnormalities are likely core to both schizotypy and schizophrenia, but they are less marked in people with schizotypal personality disorder. So just a brief summary. Early work indicates larger volume and hyperactivity of key frontal lobe regions in schizotypy. Patients with schizophrenia have lower than normal thalamo-frontal and thalamotemporal functional connectivity, but individuals with SPD are intermediate and spared. Overall, these findings are consistent with the idea that in schizotypal personality disorder, frontal and temporal lobe compensation serves as a protective factor in mitigating psychosis. So the next steps that we are taking are to look at these changes over time. So we have a new R01 that's 5 years, and we're going to be doing MRI at baseline and 18-month follow-up. We're doing all sorts of phenotyping other than that. We're doing the matrix battery, which is a full neurocog battery. We're going to be working with Cheryl Corcoran and getting some language, natural language processing done, hoping to get a supplement there to do that work. We're getting blood, and Panos is going to help us with that, and potentially another supplement. So it'll be exciting because nobody has done a longitudinal study using MRI and neurocog in schizotypal personality disorder. So we can really look at risk and protective factors over time, which will be very novel. So what about some treatment implications for this kind of work? It's important to note that people with schizotypal personality disorder do have cognitive impairments. I think I mentioned that earlier. These cognitive impairments are very much in the domain of working memory. They are less marked than people in schizophrenia. And these working memory impairments that they exhibit are mediated by the frontal lobe, and they're very consistently reported in schizotypal personality disorder. And guanfacine it's an alpha 2A agonist that acts postsynaptically in the prefrontal cortex. So this was some work that Margaret did. Guanfacine is supposed to help working memory deficits in schizophrenia and schizotypal personality disorder. So this was an 8-week randomized double-blind placebo-controlled trial for some pilot data. Margaret's now submitting an R01 to do this in a much larger sample more rigorously. But the pilot data are shown here, and they were published in the American Journal. She had a NARSAD that helped pay for this work, and it was very exciting. And basically, as you can see here, the schizotypals that got guanfacine did better on a reasoning and problem solving score from the matrix cognitive battery compared to the ones that got placebo. So we are pursuing this line of work. I'm going to shift now and talk a little bit about some of our work in borderline personality disorder and major depressive disorder and veterans at risk for suicide, because that's the other arm of our research. And a question here that's important is, what's the role of amygdala frontal regions and circuitry in emotion processing? And can we find objective measures for predicting suicidal behavior? That's something that we are looking at more recently, as Joe noted so wisely from looking at my CV. So in this area, we've done a lot of work in borderline personality disorder, kind of an understudied disorder. It's hard to get R01s to study borderline personality disorder, although Harold Koningsberg's done a pretty good job of getting a few of these. The prevalence is about 1.6% in the general population, and it's about 20% in psychiatric inpatient units. 18% of all suicide deaths are due to borderline personality disorder. This disorder heavily taxes the healthcare system, despite being understudied. It incurs high healthcare costs, about $45 $1,000 annually per patient, and it's very comparable to severe health conditions like bipolar disorder, yet understudied. So we have been studying it for decades. Larry Seaver was really a world-renowned expert on borderline personality disorder. So this disorder is characterized by emotion dysregulation, importantly. The idea that they have instability of interpersonal relationships, of their self-image, and they're very emotionally dysregulated. It's marked by impulsivity, suicidal behavior. Suicidal behavior is actually one of the 9 diagnostic criteria for borderline PD. And they have a lot of impulsive aggression. So how do we define emotional hypersensitivity? So this has been discussed by many borderline PD experts. It's basically defined as a lower threshold to detect or respond to emotional stimuli, or a higher probability of experiencing stimuli as being very emotional. That was Carpenter and Troll. Marsha Linehan has a model that discusses the biological predisposition to being emotionally hypersensitive and exhibiting long, prolonged emotional responses, which we're actually going to talk a little bit about, because that relates to habituation, in my opinion. Mary Zanarini at Harvard talks about a failure to develop appropriate and adaptive emotion regulation strategies in borderline PD. So I have this little schematic that I like to show because it helps understand some of the psychophysiology that we are using to study this disorder. So emotion dysregulation in borderline PD is characterized by this inability to regulate emotional responses, according to Marsha Linehan. High sensitivity, so they respond more to emotional stimuli, and they have a longer, prolonged response. They can't shake it off like a healthy person. So this really maps on well to something that I use to study. In addition to using fMRI, we use affective startle, which I'm going to talk about a little bit later. So the idea here is that, you know, they see an emotional stimulus, and whether you're talking about an fMRI bold response activation to that stimulus or affective startle, which we're going to talk about, the idea here is that borderline patients show this heightened peak or greater response. And then when these stimuli are repeated over here, so this is time on the x-axis, healthy people show habituation. Their bold response or their affective startle declines is the idea. But in contrast, people with borderline PD can't shake their emotions, and they show actually a bigger response, or they fail to habituate. So brain regions that are important in underlying emotion processing are numerous, as you can see in this figure from Pessoa. Red areas shown here are involved in emotion processing, and the blue regions are, you can see it looks more like circuitry. They're involved in regulation. So frontal lobe is really important in regulating emotion, but it's really the amygdala that we've targeted for looking at emotion processing. So the amygdala is important because it has a primary role in the processing and memory of emotional responses. And it's really important in determining whether something's good or bad. So the valence of a stimulus, that really taps into your amygdala. So I have submitted some papers on the amygdala, and sometimes I get a little pushback from reviewers, like, you know, what about the rest of the brain? Why are you only focusing on the amygdala? So this is my amygdala-centric slide to respond to that. The amygdala is pretty cool. The primary nuclei in the amygdala, which are shown here, the central nucleus and the basolateral complex, have been really well conserved across species, as you can see here. So here's us humans, and here's a mouse, and you know, they have an amygdala. So across species, there's a lot of evidence that the amygdala is an important component of the network of neural systems that produce adaptive social interactions. Another really key factor in borderline personality disorder. These people have a lot of difficulty with social interactions. So might it be that their amygdala is abnormal? The most consistent finding in fMRI studies examining sort of passive viewing of emotional stimuli, facial expressions, or unpleasant pictures is greater amygdala activity in these patients compared with healthy controls. And there's been plenty of reviews over the years. So I think something that makes our work unique in this area is that we, in the beginning at least, we don't do it now, but back in the day, we would trace the amygdala on everybody's MRI blind to their diagnosis using this Sobel filter that would enhance the gray-white matter segmentation. And then we would co-register these amygdala regions of interest to their bold scans and punch out basically activation in their amygdala. And then we could do these really cool mixed model MANOVAs to look at amygdala activation. So we've used the pictures from the IAPS. You see some examples here that are pleasant, neutral, and unpleasant. So it's an intermixed series of these pictures. And in the case of this study, they repeated so that we could look at habituation. And importantly, we tried to use a lot of social pictures and borderline salient pictures because that seems to be very important. And I'll touch on that a little bit later. So they were instructed to focus on the pictures and think about their meaning for you personally. So trying to make this a very personal kind of experience because that seems to be important in studying people with borderline PD as well. They had a 3-choice button in the magnet. That was mainly to keep it not too complicated so it didn't get too cognitive, but we wanted to make sure that they were awake and watching the pictures and concentrating on the task. So what did we find? Here's a picture that shows the bold activation averaged over all 3 conditions, unpleasant, neutral, pleasant pictures, keeping in mind that unpleasant and pleasant are emotional and they were matched on arousal. So equally arousing, opposite in valence. And then we had the neutral pictures. But overall, collapsed over all these conditions, what we see here is that the borderline patients, when they're shown the pictures repeatedly, they show this larger response compared to healthy controls. And actually our psychiatric control group here was a group of people with schizotypal personality disorder. So the idea of this failure to habituate, when we unpack that a little bit into the 3 different conditions, unpleasant, neutral, pleasant pictures, what we see here is that the abnormalities are really specific to the emotional pictures. So they show a bigger response to the repeated pictures in the unpleasant condition, but also in the pleasant condition compared to the other groups. And more recently, with Kim Goldstein, who's an assistant professor at Sinai and also working at the VA on some of our suicide prevention studies, we have looked at, we've sort of taken a deeper dive into this and looked at borderline patients with and without a history of suicidal behavior. Suicidal behavior being defined as a suicide attempt or suicidal ideation. And what we find is that it's really the borderline patients with a history of suicidal behavior that seems to drive this amygdala habituation deficit, which is really interesting. So the borderlines that don't have a history of suicidal behavior actually looked more normal during the unpleasant picture condition, which is where we previously reported the abnormality. All right, so brain circuits underlying emotion inhibition and regulation. I mentioned this a little while ago. So the frontal lobe is really important in regulating emotion and has connectivity with the amygdala. So there are a lot of neuroimaging studies and they generally support an emotion undermodulation model in borderline PD. And the idea here is that compared to healthy controls, borderline patients show hyperactivity of limbic regions while looking at emotional stimuli, notably the amygdala and the insula. And compared to healthy controls, they show a hypoactivity of orbital frontal cortex and anterior cingulate cortex regions in the frontal lobe. So it's like their frontal lobe isn't turning off the switch to quiet their amygdala. Now, these studies are mostly done with fMRI, but this is a shout out to Antonia, because way before we were doing fMRI and we were doing PET scans with fluorodeoxyglucose, and we were also doing challenge studies like this one where people got MCPP. We were looking at functional connectivity using FDG PET by doing correlations. So this is actually data from a paper that Antonia published that showed back in 2007, before all this fancy fMRI functional connectivity, that people with borderline personality disorder don't have a correlation between their OFC, orbital frontal cortex, which is actually, this is like an upside down brain showing the orbital frontal cortex, and the correlation with the amygdala. And so you don't see any red here in the borderline group. In contrast, people that are healthy show red because their amygdala and OFC are highly correlated. This was actually a pretty good sized group of subjects that got PET scans. This is looking at fluorodeoxyglucose or glucose metabolism in the placebo condition. So the idea here is just to show that functional connectivity studies of using FDG PET back in the day showed that there was a problem in borderline PD in amygdala-OFC dysconnectivity. More recently, this is a shout out to Harold, work has been looking at amygdala circuitry using functional MRI. And this is work from Harold showing that there's a lack of increase in the functional connectivity that's important when you're looking at those repeated unpleasant pictures, similar paradigm to what I've published on. So in borderline PD, they show actually a decrease, whereas healthy people show an increase in the left amygdala and the same pattern in the right amygdala, so it's bilateral, when looking at repeated unpleasant pictures. These are correlations between amygdala and insula. So the idea that these limbic regions are not showing normal functional connectivity. Okay, so let's look at the time, 12:36, we're good. Okay, so I want to shift gears a little bit and talk a little bit about my psychophysiology work, because I think it's kind of interesting and fun, and it's important for people to think about because it's very inexpensive to do compared to imaging work, and it's very translational. So here is a former research assistant. Just full disclosure, this is not a patient. This is just an RA showing how we can do psychophysiology in the lab. He's got his electrodes on. You can't see, but we're also measuring skin conductance from his fingertips. And he has on some headphones and he's ready to look at some pictures on the screen there. And what we're doing is measuring startle, because startle is actually a ubiquitous response, or reflex, I should say, that all mammals exhibit. So here's the 9-banded armadillo leaping into the air from the flash from the National Geographic photographer's camera. And here's a cartoon of a human opening up a can of worms and jumping into the air. So whole body startle. You can measure it in rats in the lab, and that's what is done. Or in humans, you can measure it from the orbicularis oculi muscle, as we do, and look at the startle eye blink component of the startle response. So the basic pathway involved in startle is kind of boring and to me not so interesting. But what's really interesting is that it's mediated by the central nucleus of the amygdala. So animal models suggest that the direct pathway from the amygdala to the startle circuit mediates both fear potentiated startle and sensitization of startle. So greater amygdala activation leads to greater startle response. This just shows a little bit more about how we record EMG to measure the startle blink response. And then it's important to point out that a lot of work has been done, a lot of it's done in psychology labs around the world, looking at affective startle modulation in humans. And Peter Lang has a beautiful theoretical model about this. So the idea is that you show people these pictures, which are highly evocative and emotional. Here's an unpleasant picture, a neutral picture, and at the bottom, a pleasant picture. And about 5 seconds, even 4 seconds, there's different probe positions you can use to look at the granular temporal course of this startle response. Here it's 5 seconds. So 5 seconds into this picture, you present a very brief burst of static. It's about 50 milliseconds and it's 100 decibels. So it sounds like a click sound. It elicits a blink. And interestingly, Peter Lang and others many years ago showed that if you present that startle stimulus through headphones during an unpleasant picture, you get this giant blink. The blink is potentiated, in contrast to, say, the small blink that you see when you're looking at a pleasant picture. And interestingly, the neutral picture produces an intermediate response. So this sort of phenomenon of affective startle modulation has been replicated many, many times, thousands of times in lots of papers, and it's very reliable. But back in 2007, we were the first ones to look at this in borderline personality disorder. And I took a word list that Jill Hooley at Harvard had used in a behavioral paradigm where she told borderline patients to try to forget certain words and remember other words. So I used her unpleasant and neutral words. So you can see some of the words that we presented on a computer screen to the participants, included for unpleasant condition, ugly, abandoned, useless. These are words that are almost trigger words for people with borderline personality disorder. And then we use neutral words. And we presented the words for 6 seconds on a computer screen, and we told the participants to look at the word, read the word quietly to themselves, and think about the meaning of the word for them personally. And then we drop startle probes in. It's also important to note that we measure baseline startle. So here's an example over here, no picture. There's just a startle probe presented alone. That allows us to measure the amplitude of the eye blink response to just a startle stimulus. And all these affective startle measurements are relative to baseline for each individual. And then we drop the startle probe into some of the word pictures. And what did we find? Basically, we showed that compared to healthy controls, people with borderline personality disorder showed much greater, significantly greater amplitude of their eye blink response during unpleasant words compared to the healthy controls. They didn't differ for the neutral words. But really, most interestingly, we also asked them about each of the words and had them do a self-report of valence on a 9-point scale, which is sort of a standard tool that's used in affective startle research. And what we found was that the borderline patients had a dissociation between their psychophysiology, shown up here, and their self-report. They basically said that the unpleasant words weren't really that bad relative to the healthy controls. So they thought they were bad compared to neutral words. It did show this significant difference on their support compared to the healthy controls. So a mismatch between their physiological and behavioral emotional response, which is interesting and pretty much characterizes one of the problems people with borderline personality disorder have, is that perhaps they're not good at describing their own emotions. So we published that paper in Biological Psychiatry, and I was happy to see some years later that a group in Germany replicated this finding using affective startle. And they took it a little bit of a step further by using scripts that were specific to the person, and they had neutral scripts, and then they had sort of unpleasant scripts, and then they had these rejection abandonment scripts, which are very borderline PD specific. And they showed that they differed, the borderline patients differed, from healthy controls only for the rejection abandonment condition, interestingly, and not the other conditions. So more recently, we have started to use those same images to look at fMRI. And we've also used them at the VA. So this was actually startle work we did at the VA in a pretty good sized sample that was transdiagnostic. So you can see here, there were 4 groups: ideators that were passively ideating, active ideators, people that had a single suicide attempt, and multiple attempters. So 4 groups, and they were age and gender and education matched. And we wanted to look at whether they differed in affective startle and whether affective startle increased in terms of the unpleasant condition as suicide risk increased. In other words, are the multiple attempters showing even more exaggerated affective startle to unpleasant pictures compared to the other groups? And here are the findings. And basically, this is what we found. These are the 4 groups. This is the unpleasant picture condition, and the affective startle response was greatest in the attempters, the multiple attempters, I should say, compared to the other groups. The groups didn't differ for neutral or pleasant. And then, interestingly, when we looked at early versus later trials, we found this same habituation abnormality that I've been telling you about. So it was the multiple attempters that actually showed a bigger affective startle response on the later trials relative to the other groups. So this failure to habituate in terms of their affective startle response over time to the unpleasant pictures. Interestingly, the groups didn't differ on self-report, important to know. We did some self-reports of emotion regulation and showed that greater startle during the unpleasant pictures was highly associated with greater emotion dysregulation. And interestingly, we, in this sample, were able to look at baseline startle, because this was a cross-sectional study, and whether, because they went in to a treatment study following the psychophysiology study that I did with Marianne Goodman, we were able to look at whether affective startle was a predictor of a suicide attempt. And actually, this was a small group of 19 veterans that were in the placebo condition, or I should say a treatment as usual condition for Marianne Goodman's DBT, dialectical behavior therapy treatment, randomized clinical trial of DBT in veterans at risk. And what we found was that it was the veterans at baseline that showed bigger startle blink during unpleasant pictures that had a suicide attempt at their 12-month follow-up. So really interesting and promising data, and we're now finishing up a merit that was based on this preliminary data, and we're going to be looking at that in a much larger sample and trying to replicate the idea that perhaps affective startle could be a biomarker for suicide. We have more recently also looked at affective startle in borderline patients using the same paradigm as we did in the veterans. So this is unpleasant, neutral, and pleasant pictures. And we find, again, that the borderline patients compared to healthy controls and people with schizotypal personality disorder show this exaggerated affective startle response only in the unpleasant condition. And they don't differ from the other groups in the neutral or pleasant condition. And again, we find that the borderline patients show this abnormality in their habituation to the unpleasant pictures. So this, I put it together on one slide. This affective startle work really looks similar to the work we published previously in the amygdala. So I think it's going to be really important to try to assess these measures together and see if they are actually correlated, because it appears that they might be. So in terms of treatment implications, and I'm getting ready to finish up here. Treatment implications. I think of this work that Marianne Goodman did, where we, I think this was part of her career development award, but I can't quite remember, Marianne. But we looked at amygdala activation in borderline patients that went into her DBT study. And heroically, she gave them 12 months of dialectical behavior therapy, which is not easy. And interestingly, their amygdala did quiet and go down following DBT. And we didn't have a patient control group here, but we just compared them to healthy controls who were very stable over time. And similarly, the patients showed a decline in terms of their emotion dysregulation. So they got better in terms of regulating their emotions over time as well. So I think it's going to be important to study biological predictors of treatment response like this. I'm hoping that we can look at some of these measures like affective startle and measuring the amygdala using fMRI pre and post treatment in some of our veterans at risk for suicide. All right, so some conclusions and implications from some of this work. I showed you that borderline patients have abnormalities in terms of hyperactivation of their amygdala and exaggerated affective startle to unpleasant pictures. We're gonna try to see if these responses are correlated. I think that'll be important going forward. Affective startle is a promising biological end of phenotype for suicide risk potentially. We really need objective measures of suicide risk. We don't have any. So we are working on this. Our finding of increased suicide risk being linked with this startle pattern suggests that amygdala hyperactivity is associated with suicide risk. And we're currently working on a couple of VA merits, looking at psychophysiology and doing fMRI. We now do fMRI scans at the VA. We have a 3T scanner, which is great. And I do think these findings match or align with Tom Joyner's work talking about two general categories of psychological risk for suicide. The idea of a propensity to intense psychological pain. Maybe this is something genetic or environmental that borderline patients suffer from. And then this idea of dysregulated impulse control. So the idea that dysfunction within amygdala circuitry may dysregulate emotional expression and experience, which then gives rise to this overt suicidal behavior and suicide risk. I want to thank the VA and NIMH for my funding to do this work. And then I'll stop here, but I want to say that we are recruiting for this study that we're doing. Phil Zesko and I are working on this R01 study at Sinai. It's a longitudinal study. We're looking for healthy controls, people with schizophrenia or schizotypal personality disorder. We are advertising, but I thought I'd put this out there for anybody who might be interested or have a patient that they would like to refer. You do get an MRI report. You get a very deep phenotyping, all sorts of clinical assessments and cognitive testing. And you can call Alensera if you're interested or have a referral. And I'll stop there.
Well, thank you, Erin, for a great talk. And it's open for questions. You can either type in the Q&A or raise your hand, and maybe I'll get started. When you go back to the first part of the talk, and you're talking about the hyperactive prefrontal cortex, right, as perhaps protective in SPD, Is there an opportunity for intervention there? Are people trying to, for example, do transcranial magnetic stimulation in frontal areas and seeing if it provides some, relief in first break or in the prodrome?
You're talking about in schizophrenia?
Yeah.
Yeah, yes. The answer is yes, and I don't know much about that work.
All right, well, I got my answer, so.
Yeah.
Well, I mean, I think, you kind of ended it, well, you ended part of it by talking about treatment implications and
Right, yes. So yeah, so like Margaret's working on Guanfacine. There are people doing transcranial stimulation. I mean, that makes a lot of sense. Not really up on that work. I probably should be more up on that work.
Well, Cheryl Cochran has just said that TMS, the transcranial magnetic stimulation to dorsolateral prefrontal cortex, has improved cognition in schizophrenia patients.
Well, there you go. I mean, it makes sense. And it could be interesting. I don't think anybody's looked at that in schizotypal personality disorder. Or how about clinical high risk, Cheryl?
Exactly, in the prodrome, wouldn't that be interesting?
Yeah.
All right, Cheryl, wait.
Work to be done, work to be done. Got to get on that, Cheryl.
All right, additional questions? Like I said, you can chat or, okay, Cheryl's answering again. Prodrome. No known TMS and schizotypal that I know of. Not everybody can see the chats, I think. I'm not sure, so that's why I'm reading them. Prodrome is equivalent to TMS to cerebellum for negative symptoms. Okay, I don't even know what that means. So I'm going to allow Cheryl to talk. Cheryl, you're allowed to talk now.
The equal sign is wrong. It should be, there have been TMS studies in clinical high-risk patients, I think, and schizophrenia to the cerebellum for negative symptoms. There hasn't been too much TMS in clinical high risk. I would love to do TMS in clinical high risk. I used to do a lot of TMS at Columbia for cognitive neuroscience. So would be happy to work on that with you, Erin, for schizotypal and CHR and schizophrenia.
Who's doing TMS at Mount Sinai?
You mean at Sinai?
Yeah, like in general, like is there somebody doing it? I don't even know.
I believe there's
this is why we have these virtual talks, is to network.
They're using, of course, therapeutic TMS for treatment of depression. But TMS can be used also as a really cool probe for cognitive neuroscience studies because you can establish causation. So we used to replicate all kinds of cognitive deficits transiently in healthy individuals at Columbia, I guess for sport, but seriously to figure out the underlying circuits.
So I have a question from Anitha. Erin, I can read it or you can read it.
Okay, I see it actually. Curious about your thoughts on Joyner's question that suicidality and related affective elements be considered a separate syndromal category or disorder. Like a separate category or disorder in the DSM? I'm not exactly sure what the question is, but
and Anita, if you want to, I've allowed you.
I mean, can she
clarify? I know that Joyner has suggested that there's enough that's separate and different about the affective elements of suicidal individuals that there be this sort of consideration of acute suicidal affect and sort of looking at those as a separate category or even a separate disorder in the DSM moving forward, obviously.
Yeah, I mean, the whole RDocs idea, that we've all been sort of, in my opinion, struggling with. I don't know. I think it's an interesting idea. I don't know. I think it's hard. Like in our VA study where we had 108 veterans at low to high risk for suicide. If I look at their disorders, they range from MDD to a couple of them had bipolar disorder. So all kinds of mood disorders. For me, it's hard to parse it out. I like more homogeneity, but
yeah, I mean, and I don't have too much more information on it. I had heard him presented at American Association of Suicidology some years ago. And I'm not sure if he's done any more with that since, but I was just curious about your
yeah,
thoughts. So thank you.
yeah, I think it's an interesting idea, but it's messy in reality.
So maybe we should wrap up. I see that Cheryl had one more comment to make. Nice talk, by the way. That's a quote. There should be a comma. Nice talk, comma, by the way. Clear and covered well a few
Oh,
different areas of
thank you.
research. So
Yeah,
Erin,
you
it was a
cover a
great
lot.
talk. It was a great talk, and thank you for doing it. And like you said, these are opportunities for people to begin to think about collaborations and hear what each other's doing and kind of figure out exciting things to do together. So thank you for doing this.
Yeah,
And,
yeah. Plug for the MyRec fellowship program as well. You know, it's a great place to come and do a fellowship, and it's a great place to apply for career development awards. We have a really good track record of people getting career development awards at the VA, and they're similar, if not the same, as the ones you get from NIMH. So just a little hot tip there.
yeah. Yeah, there's been a great, like you said, a great success record, and I think the salary can be higher than the NIH.
It might be higher. Isn't that amazing? I don't know. Something to think about if you're on here and you're a resident.
Well,
All
thank
right.
you. Next talk will be in 2 weeks, and I tried to quickly pull up who's presenting, but I'm not organized. So I'll see you all in 2 weeks. It'll be exciting, whatever it is.
Thanks, Joe.